In this video, we will discuss about the relationship between galectins and the immune response. So we will start with the T cell activation. So this T cell activation, the signal for the T cell receptor is strong when the major histocompatibility complex interacts with this CD18 directly. However, when the signaling pathways such as interleukin 10 or MGET 5 got triggered, then this will lead to weakening of signals, and in this weakening of signals this galectin 3 also plays a role, as it will interact with the increased N-glycan branching of the CD18 and other cell receptor, thereby reducing this CD8 interaction with the major histocompatibility complex, and this results in weakening weak T cell receptor, T cell activations, weak signals for the T cell receptor, or in other words, uh, it will decrease the T cell activations. And in terms of intracellular galectins, this galectin 3 interact with this helix and thereby this uh, reduce this CD8 interaction with the major histocompatibility complex and decrease the T cell receptor signals, whereas this galectin 9 will increase the interaction of this will increase the signal for the T cell receptor through some intracellular interactions that are yet to be identified. So this is how T cells and T cells activations is related with the galectins. Now let us discuss about T cell contraction. So what happens in this case? A study has been conducted where the mice are grown with the mice are grown that does not have or mice are grown with the galectin 1 to 8 9 knockout, and in that case it has been observed that T cell contraction has been found, and when these galectins are reintroduced, uh, that is 1 2 3 8 and 9, then it will trigger the apoptosis of the cells. So this is one of the observations that has been found about T cell contractions in the mice and its relationship between galectins. And another intracellular relationships about galectin is that it will prevent the apoptosis by reducing this site C leakage from the mitochondria with the help of this BCL2 binding BCL2 interact with the mitochondrion. Thereby, the leakage of site C will be reduced or decrease in the cytosols of the cells, and this will the apoptosis through this site C. The triggering of this apoptosis through this site C will get reduced. So this is how the galectin 3 is interacted with, is related to the apoptosis, or we can say the T cell contraction. So, and next one is about the B cell differentiation. So what happens in this case is that B cells, in the absence of this galectin 3, what happens, the B cells will be able to differentiate himself into the germinal center B cells, and this can produce antibodies such as Ig2 and Ig3, and this finding can help us to develop autoimmune response. This finding can give us an idea how this autoimmune response will get triggered within the disease patient. So what happens in this case is that B cells are able to produce their what happens in the normal case is that B cells are able to produce their own galectin 3 that can prevent these B cells to differentiate themselves into the germinal center B cells. So this is one of the relationship of B cells with the galectin 3, and it has also been found out that help in the other galectins this prevent other galectins in the absence of galectins, these B cells can be differentiated into the plasma cells also, and these plasma cells can also produce the antibodies such as Ig2C or Ig3. So this can cause the autoimmune response. So this in these B cells, these galectins helps in this case the galectins helps in preventing the undesired differentiation of these B cells to prevent the autoimmune response. So, and the next part is that of about B cells activation in different cases, such as naive B cells and germinal B cells. In the naive B cells, what happens is that the galectins helps in interacting CD44 and CD45, and thereby weaken the signal of B cell receptors. Through this SHP1 interaction with CD45 and this CD44, this CD22 and CD45 co-interaction because of the galectin will decrease or weaken these B cell receptor activations in the case of naive B cells, thereby prevent the thereby helps in reducing the immune response of these B cells because of B cells. However, in case of germinal center B cells, because of the different carbohydrate antigen, what happens because of the different carbohydrates that are present on the B germinal center B cells, the galectins will not be able to interact with them, and thereby does not cause weakening of the signals, and uh, thus in the case of germinal center B cells, what happens is that the antigen interacting with the B cell receptor will cause a strong signal, and this activated B cells or germinal center B cells will trigger immune response against those antigen carbohydrate. So this is how these naive B cells are controlled uh, from being matured into the germinal center B cells, and this is how we can say the immune response controlled in a manner that it will prevent undue maturation of naive B cells into the germinal center B cells. If you need to understand this galectins and immune response in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.